Okay, we're going to get going. It's a beautiful day outside. Feels like summer, 80 degrees. So today our first speaker is Perez Hussain. He got his PhD in India, subsequently was a postdoc at the Swiss Institute for Research on Cancer in Lausanne. And then he saw the light and came to NCI. And he was also on the faculty at the University of Maryland School of Medicine in Baltimore, and then a staff scientist, and now he's a tenure track investigator. He's in the Lab of Human Carcinogenesis, head of the Pancreatic Cancer Unit. The title, Inflammation and Cancer, an Ancient Link with Modern Evidence.
Okay, thank you, Terry, for the introduction. So as you can see, my title is a vast title, and inflammation and cancer. It's such a huge field and highly complex, and it requires the coordination of a number of events that occur during inflammation, especially in chronic inflammation. So what I'm going to do, I'm going to give you some examples of the events that occur during chronic inflammation, and then focus on few chronic inflammatory diseases and some cancer types. And I give you a little bit more details, just a little bit focusing on inflammatory mediators, and one is the nitric oxide and another is the macrophage migration inhibitory factor. So with that, let me start with this slide. I just did a search on inflammation and cancer. And in last 10 years, there are 37,201 articles that are published. So this concept is not new, and a lot of work has been done and still going on to dissect the mechanisms that are involved in this process. So more than 100 years ago, Rudolf Virchow noted the presence of leukocytes in neoplastic tissue and made a connection that there is an association between inflammation and cancer. And then all the recent evidence based on experiments, clinical studies and epidemiological studies have supported this hypothesis, and the links between inflammation and cancer, they may have some implications in prevention and treatment of cancer. So this is one of the largest epidemiological studies that showed that aspirin use reduced the risk of cancer death, as you can see here, and they enrolled about total 23,000 cases, 13,000 with taking aspirin and 10,000 as control. So this busy slide is just to tell you that there are association between chronic inflammatory diseases and several different types of cancer. And these chronic inflammatory diseases can be either inherited. There is a familial component associated with these diseases. For example, hemochromatosis, iron deposition in the liver, hereditary pancreatitis, where you see a mutation in the trypsinogen gene that in fact enhance the development of pancreatic cancer. Then ulcerative colitis and Crohn's diseases, these are inflammatory bowel diseases that enhances the risk of colon cancer. Or it can be acquired through infection with viruses, bacteria, and parasites. For example, hepatitis, cis virus B and C, that enhance the risk of liver cancer, Helicobacter pylori, and pelvic inflammatory diseases enhances the risk of gastric and ovarian cancer. And then parasites, for example, Schistosoma japonicum or hematobium, and then liver fluke infections. And then also the exposure to chemicals and physical and metabolic changes can also enhance the development of cancer. And one of the example of metabolic diseases is obesity, which I will be showing a couple of slides on. So any stress caused by infection or injury can induce immune responses, both innate and adaptive immune responses that induces inflammation. And during inflammation, a number of different factors, including cytokines, chemokines, free radicals, prostaglandins, growth factors, MMP, matrix metalloproteinases, and so forth are produced. And these factors interact with both epithelial cells and stromal cells and can contribute to several events that can be tumorigenic. For example, DNA damage, including mutations, protein modification that can lead to either a stabilization of protein, for example, tumor suppressor p53, or alterations of different tumor suppressor genes, or activation of oncogenes that can lead to proliferation. And there are many evidence that microRNA expression can be regulated to some extent by different cytokines. Then DNA and histone methylation, and also neoangiogenesis that is required for tumor progression. So we see that there are strong tumor stromal interaction that is mediated by several of these factors, especially chemokines and cytokines, and that can enhance progression or metastasis. In adaptive immune response, on one hand, it can contribute to tumorigenesis to some extent, for example, enhancing T regulatory cells that can suppresses anti-tumor immunity, and also humoral immune response that can enhance chronic inflammation. On the other hand, it can also induce cytotoxic T cell activity and tumor cell lysis. So this is the oversimplified explanation of inflammation mediated generation of several factors that might contribute to tumorigenesis. So one of the important events is the generation of several reactive species, for example, reactive oxygen species or reactive nitrogen species, and then their combination. So reactive oxygen species, for example, hydroxyl radical, and please stop me at any point too if I'm not clear in my explanation. And superoxide anion and nitric oxide, and then nitric oxide can interact with oxyradical or superoxide anion to generate more reactive peroxynitrides and nitrous anhydride. And these reactive intermediates can induce DNA damage, including point mutations, either directly or indirectly through initiation of lipid peroxidation and generation of malondialdehyde and 4-hydroxynonenal, and the production of ethenoadducts. And these ethenoadducts have the capacity to induce mutations in DNA and genes. In addition, it can also modify proteins that are involved in DNA repair and apoptosis. So several evidence suggests that where inflammation works, does it contribute in the development of the tumor, or the progression of tumor, or the metastasis of tumors? So evidence showed, as described here in a very good review by Michael Curran et al., that it can contribute in all the initiation, promotion, and metastasis. During initiation, these species and cytokines, as I discussed, can induce epigenetic alterations and mutations in the pre-malignant cells. And once the transformation takes place, the cancer cells start producing these factors, for example, chemokines and cytokines, and there are feedback loops that contribute to survival, growth, growth and also neoangiogenesis. And then during metastasis, these factors can induce epithelial to mesenchymal transition, survival, and invasion and motility because of the epithelial mesenchymal transition. There are also inflammation effect can be categorized as in different stages. So the chronic inflammation due to infection and autoimmunity, and the inflammation caused by dietary and environmental exposure can induce mutations, genomic instability, tumor promotion, and angiogenesis. And then also the tumor itself can induce inflammation in the tumor cells and in the surroundings that can lead to tumor growth, survival, genomic instability, angiogenesis, immune suppression, but can also lead to cancer cell killing because of some of these factors. And then therapy can also induce inflammation. At one hand, it kills cells, it produces necrosis, but that leads to the development of therapy-resistant cells and tumor reemergence due to the therapy, but can also at the same time enhance the antigen presentation and lead to cancer cell killing. So a couple of years ago, Steve Hursting, who used to be at NCI, have described that obesity is a metabolic syndrome, and what happens that if you intake high energy and physical activity is less, that can lead to chronic positive energy balance that can lead to obesity, which is a metabolic syndrome and can lead to enhanced growth factors and cytokine that are mentioned here that can aid in the growth factor signaling, including PI3 kinase, AKT, mTOR pathway, enhances vascular perturbation and inflammation, leading to the induction of oncogenic signaling pathway, for example, NF-kappa B and COX-2, that can enhance the cancer risk and progression. So several years ago, it was shown that, in fact, adipocyte during these metabolic syndrome start behaving like macrophages, in fact. You see the macrophage infiltration in the adipose tissue, adipocytes, and both of them start generating some of these factors, for example, cytokines, and can aid to the increased risk of developing cancer. So a number of different pathways, oncogenic and tumor suppressive pathway, can be affected during chronic inflammation through the production of cytokines and free radicals, as I mentioned earlier, that can lead to DNA damage and enhancement of NF-kappa B pathway. It can also lead to stabilization of p53 pathway through ATM by causing p53 phosphorylation and stabilization that can lead to cell cycle arrest, DNA repair apoptosis, and can also inhibit RB pathway. So it's a complex situation, and it all depends on the timing and the biological context, that which way the chronic inflammation and its induced pathway will go. So one of the inflammatory mediator is nitric oxide. So I don't know if who all of you have attended David Wing's lecture, and I don't know how much he has talked about nitric oxide. So if I repeat that, please excuse me, but I will be as brief as possible, just giving you just a little background that nitric oxide is one of the free radicals. It is generated during the conversion of L-arginine to citrulline, and this reaction is catalyzed by a family of enzymes, that is nitric oxide synthases, that are categorized broadly into two types: One is NOS1 and NOS3. They are constitutive. They are calcium dependent. And NOS2 is inducible, that is induced during chronic inflammation and produces a very high level of nitric oxide. So nitric oxide immediately interacts with molecular oxygen or superoxide anion, which I mentioned briefly earlier. It can lead to nitrosation, deamination, oxidation, and nitration, has been shown by many different published work, and that can lead to DNA damage, mutations, protein modifications. That is one pathway how nitric oxide can be tumorigenic. So NOS2, which is the induced form, it is induced by several different stimulation during inflammation. Cytokine production, beta-catenin, TCF4 can in fact bind to NOS2 promoter and enhances NOS2 expression and NO production. Several endotoxin, microRNAs, NF-kappa B, hypoxia, and oxidative stress. One of the interesting pathway of nitric oxide interaction with the tumorigenesis has been shown by its feedback inhibitory loop with p53. So what happens, that was shown in Kurt Harris's lab by Kathy Forrester and Stefan Ames, nitric oxide can damage DNA, lead to the stabilization of p53, and p53 trans-represses NOS2. So there is a negative feedback loop. And I'm explaining this to you now because I will give you one example of chronic inflammatory diseases where we see this event. So that chronic inflammatory disease is ulcerative colitis. This is the normal colon when you do the endoscopy. And here is the lesion which you find in ulcerative colitis. And that lesion over the long period can develop ulcerative colitis associated cancer. And this is one of the studies that showed the increased risk of developing colon cancer in chronic inflammatory disease, ulcerative colitis. So this slide, these are all sections from ulcerative colitis patients. This is normal, and then you see NOS2 immunohistochemistry. Here you see a lot of NOS2. You see an association with the phosphorylated serine 15 staining in the p53 gene, and also p21, which is the target of p53, and also MDM2. So what this slide is telling you, that in the tissue of chronic inflammatory disease ulcerative colitis, you see increased expression of NOS2, and you also see increased expression of phosphorylated serine 15 site or p53, which is important for the activation of p53 and p53 functions. And these are the downstream target of p53 that you can also see. So this shows an association between NOS2 and p53 stabilization. So simply we can say that in ulcerative colitis, NOS2 produced nitric oxide can induce DNA damage, leading to the p53 activation through phosphorylation, and that can aid in the anti-carcinogenic effects, for example, growth arrest, DNA repair, apoptosis, antioxidant, and nitrosative stress. But this is not this simple, because another hypothesis will be, as I mentioned, that nitric oxide can induce DNA damage and lead to the mutations, point mutations. As you know, that it's the p53 is highly mutated in several different cancer that can derail p53 function, and in fact lead to acquiring new oncogenic functions. So we tested that hypothesis, that does this DNA damage induce p53 mutation in these chronic inflammatory diseases? So for that, they have not developed cancer. So you have to have a highly sensitive methodology that can detect p53 mutation before the clonal expansion of those cells in the tumors. So we used a highly sensitive methodology, and I'm not going to go into that methodology. That was done a long time ago. But we were able to identify one mutated p53 allele out of more than 5 million sequences. And by that, we identified increased p53 mutation load in several chronic inflammatory diseases, ulcerative colitis, hemochromatosis, and Wilson's disease, and published a series of papers on this that these diseases, they do have p53, increased p53 mutational load before they develop into tumors. So to summarize, nitric oxide can activate, on one hand, the protective p53 stress response pathway, but on the other hand, it can also induce p53 mutations and procarcinogenic effects, for example, genomic instability, decreased cell cycle checkpoints, apoptosis and DNA repair, and pro-oxidant and nitrosative stress.
So these, the source of NOX2 derived NO, you're saying macrophages have primary, within the tumor cell?
Both.
Okay.
Both. When you stain, yeah. When you stain the cancer tissue, not only colon but also lung, you see heavy expression, strong expression of NOS2 inside the tumor epithelial cells, but also on the macrophages and other inflammatory cells. So pancreatic cancer. So pancreatic cancer is one of the deadliest cancer, and it's the fourth leading cause of, in fact, third leading cause of death due to cancer now. And one of the major factor is the chronic inflammation. When you see the chronic pancreatitis patients, and if they have hereditary chronic pancreatitis, their risk of developing pancreatic cancer is more than 54. Even if they don't have hereditary pancreatitis, they just have chronic pancreatitis, they are at high risk of developing pancreatic cancer. In addition, when you look into the tumor, pancreatic tumor, it is characterized by huge stroma, and you see a small tumor mass within huge stroma, which is highly inflammatory and active. And it is shown that that stroma, which is called desmoplastic stroma, is very important for disease aggressiveness and also therapeutic resistance, because they do not allow drug to go through those stroma and act upon tumor cells. So this is desmoplastic stroma, which I just mentioned. You see constitutively active NF-kappa B signaling. You see constitutively activation of COX-2, NOS2, and also several different inflammatory cytokines that you can find in pancreatic cancer. This is a study that was published in New England Journal of Medicine that showed the increased incidence of pancreatic cancer with time in the patients with pancreatitis. So this is what I told you about. If you look at this H&E section here, you see huge stroma. These are the tumor glands here. This is huge stroma, inflammatory cells, fibroblasts, and this is called desmoplastic stroma. And there is a strong tumor-stromal interaction. Stromal cells contains fibroblasts, stellate cells, endothelial cells, and so on. Extracellular matrix, for example, collagen, laminin, fibronectin. And there are a number of different stromal pathways that are activated to mediate these tumor-stromal interaction. This is the schematic representation showing how, when normal epithelial cell is transformed into pancreatic intraepithelial neoplastic lesion, which is the precursor lesion of pancreatic cancer, and when that transforms into pancreatic ductal adenocarcinoma, you see a stepwise accumulation of inflammatory changes, that how inflammatory cells are migrating here epithelial cells from cuboidal to columnar shape, And then here you can see breaking of basement membrane and invasion of tumor glands into the stroma. Additionally, KRAS is highly mutated in pancreatic cancer. And about more than 95% of the pancreatic cancer tumors, you see mutation in KRAS genes. So all the KRAS mutation is not able to generate tumor development. It has to be maintained at a pathologic level. So in order to maintain the pathologic functional level of KRAS, KRAS mutated cells induces inflammation, and during inflammation, reactive species are produced, for example, prostaglandin E2, and that is important for maintaining the pathologic level of mutated KRAS. And this was a very good study. I thought I will present to you from Craig Logston lab at MD Anderson that was published in Journal of Clinical Investigation. So you can see that how inflammation is aiding in maintaining the oncogenic or pathogenic level of KRAS in pancreatic cancer, where it is frequently mutated. So I talked about NOS2 in ulcerative colitis and colon cancer. Let me give you one of our lab example. What we did, we analyzed the human pancreatic tumors and then simply did the QRT-PCR for NOS2 expression. And what we found that the patient who had a higher expression of NOS2 had really poorer survival as compared to those patients who had low NOS2 expression. And the patients were categorized into high and low based on the median value of NOS2 expression. Then when you do the immunohistochemical analysis on these patients, you see increased NOS2 expression in the tumors, but in the non-tumor adjacent tissue, these are the normal ducts, you don't see that much of NOS2 expression. So pancreatic ductal adenocarcinoma, I must mention, that is the most common form of pancreatic cancer, and more than 90% of the pancreatic cancer are represented by pancreatic ductal adenocarcinoma. So this simply shows an association. Is nitric oxide doing anything in development or progression? So in order to answer that question, we use a very good mouse model. So this mouse model is known as KPC mouse model because it has mutated KRAS, mutated P53 that is specific to pancreas, and it is activated by Cre recombinase. So these mice that has activated KRAS, activated mutant P53, develop pancreatic cancer that mirrors exactly the development and progression of pancreatic cancer in the humans. This is simply a H&E of the sections of this pancreatic ductal adenocarcinoma. So we asked the question that is nitric oxide important for disease aggressiveness? So we knocked out NOS2 gene from these mice, and we found that the NOS2 deficient mice had enhanced median survival as compared to the NOS2 wild type mice. So I won't go into the detail. This was just published a couple of 2 months ago, and mechanistically when we investigated that what nitric oxide is doing, so one of the pathway that we reported is that nitric oxide was inactivating forkhead box family of transcription factor type O or FOXO through the phosphorylation. So FOXO has a several tumor inhibitory role. For example, it enhances apoptosis, cell cycle arrest, DNA repair, and immune regulation. When it is phosphorylated, it loses its function and it is inactivated. So that's what we reported, that nitric oxide through initiation of ERK pathway inactivate FOXO in this model. So not only NOS2, but also NOS3 or endothelial NOS. So earlier, Chris Counter at Duke have shown that, in fact, a smaller amount of nitric oxide that is generated by eNOS or NOS3 also plays a role in pancreatic cancer. And when he knocked out eNOS from the same model which I described you, what he found that the precancerous lesions was statistically different, like there was much less precancerous lesion in the NOS3 deleted mice as compared to the NOS3 wild type mice. So, but he did not see a difference between in survival of the mice that died due to pancreatic ductal adenocarcinoma. However, there was difference in the initial precancerous lesions. So in our study, we were trying to see that. So let me tell you something, a little bit of background in pancreatic cancer. So only 15 or 20% of the pancreatic cancer patients are diagnosed early, a stage 1 or a stage 2, and then you can do surgical resection and you will hope that, okay, now everything is removed, it is a stage 1, surgical resection margin is 0, patients should live like 10 years or 15 years. But more than 80% of the patients, even in a stage 1, develop recurrence and they die. Median survival is only 2 years. So we were asking the question not about nitric oxide, but in this study we were asking the question that a stage 1 patient or a stage 2 patient, many of them who die early, but some survive longer to 5 years. Are there any difference, molecular difference in these two patient population who come from the same age group, nice resection? What is the difference? So during that study, when we did the transcriptomic analysis in a small cohort, we found that there were many differentially expressed genes, and one of the genes that was prognostically significant was nostrin. So if you have low nostrin, you had poorer survival. If you have high nostrin, you have better survival. That means it gave an indication that nostrin may be tumor inhibitory, but we don't know that. So what is the function of nostrin? One study, so what is nostrin? It's nitric oxide synthase traffic inducer. So nostrin was described to bind to eNOS from the cell membrane, bring it inside, and reduce its function and reduce level of nitric oxide production. So sequestering eNOS was the function of nostrin. So we said, okay, that is interesting. So what we did, we checked that, okay, what nostrin that is associated with survival in pancreatic cancer patient is doing here. So what we found that nostrin was inhibiting the eNOS phosphorylation, not only sequestering, but inhibiting the eNOS phosphorylation, because eNOS phosphorylation is important for the generation of nitric oxide. So nostrin inhibited eNOS phosphorylation, reduced nitric oxide. And it was regulated by another microRNA, miR-221, that was described to be highly differentially expressed microRNA in pancreatic cancer as compared to the controls. So there was a nice regulatory loop between nostrin, nitric oxide, and then nitric and miR-221. And so we concluded that nostrin is a negative regulator of disease aggressiveness. But the point here is that nitric oxide, both produced by NOS2 and also produced by NOS3, is important for pancreatic cancer development and progression. That is the point I wanted to make. One other cytokine I would like to mention a little bit more in details, that is macrophage migration inhibitory factor. And macrophage migration inhibitory factor, it's called because when it was discovered as a factor that was secreted by T cells, but it inhibited the random migration of macrophages in vitro. So it doesn't mean that MIF always in vivo inhibit the migration of macrophages, but this name was given based on this in vitro observation. But in fact, MIF can also act as a chemoattractant of macrophages, but I won't go into that details. But I just mentioned that how the MIF came into existence. So this is a pro-inflammatory cytokines. Yes. GIF. Yes. Yes. Glycosylated inhibitory factor. Exactly. It was described as a pro-inflammatory cytokine. It expressed in epithelial and a number of inflammatory cells. And to answer your previous question, it also expressed by a number of tumor cells. And so it regulates immune and inflammatory responses, increased expression in many tumors, including pancreatic cancer. It activates several oncogenic signaling pathways, NF-kappa B, AKT, ARK1, and 2. It enhances enzymes like COX-2, NOS2 that enhances or catalyzes the reaction that produces reactive species, and it inhibits, in fact, p53 function and RBE2F pathway. So based on these and several other functions of MIF, it was described as a inflammatory, as a link between between inflammation and cancer. So I thought that I will talk about MIF. That is also one of my interests in pancreatic cancer research. So we started by first doing immunohistochemical analysis of tumors in several cohorts of pancreatic ductal adenocarcinoma. You see increased expression of MIF here. And again, the Kaplan-Meier survival analysis. If you have high MIF, you have very poor survival. If you have low MIF, you have better survival. As compared to the high MIF survival group, pancreatic cancer, never better survival. This is the multivariate analysis to show that MIF is a candidate independent predictor of prognosis in pancreatic cancer. And then it is very important to validate these observations in several independent cohorts, which we did. You can see in 3 different cohorts and 1 publicly available data set in a recent data set that was published in Nature Medicine, that we took their data and checked for MIF expression, and you can see the increased MIF expression was associated with poor survival in pancreatic cancer. So that is again an association. We don't know what MIF is doing. That was simply an association with survival as a biomarker. So we did a study where we expressed MIF in the pancreatic cancer cell lines and did the orthotopic xenografts. You can see that MIF-expressing tumors were larger in size and also tumor weight, and they also had higher metastasis in distant organs. Most interestingly, when you look at the histology of these 2 tumors, so MIF-expressing tumors were always poorly differentiated in these orthotopic models, and the control tumors were poorly to moderately differentiated. We did the gene expression array analysis. You see that MIF induced a change in the global gene expression profile, and again, I will not go into the details of this study, but what we saw that it induces changes in the EMT-related genes. For example, MIF-expressing tumors had reduced ecadherin, increased vimentin, and ZEB1. So we wanted to see whether it's what is going on in here. In the meantime, recently, after we published that data, a paper came out in Nature Cell Biology, and we were very happy to see that paper. What it showed that that exosomal MIF is very important for preparing a metastatic niche in liver for the pancreatic cancer cells. And what it said that the surgically resected patient who had increased exosomal MIF has much higher chances for metastasis into liver. So these findings suggested that exosomal MIF primes the liver for metastasis and may be a prognostic marker for the development of pancreatic ductal liver metastasis. So we went back to our human samples, and we found here too, like in mouse, that low MIF had well-differentiated tumors here, high MIF poorly differentiated tumor. So we took a integrative transcriptomic approach to answer the questions that are there molecular distinction in these two tumor groups, and what are the mechanistic and functional role of MIF in tumor progression. So without going into the details, I will show you the schematic. This just came out in August issue of Cancer Research, where we showed that MIF induces PI3 kinase AKT signaling, which was known before, but what we described that this signaling enhanced the expression of MIR301B, which inhibited a novel tumor inhibitory gene, tumor suppressor NR3C2. The role of NR3C2 we described as inhibitor of EMT. So when 3OB is increased, inhibits NR3C2, it enhances EMT, and that can enhance the progression. What we don't know right now, and we are working on, that what is here, what is leading to the increased expression of MIR301B. So to bring this idea further towards therapeutic model, what we did, that we again used the same mouse model which I described. We deleted MIF, and we found that when you delete MIF here, that is MKPC, you see much longer survival as compared to the mouse that has wild type MIF. So that was the study. Now what we are doing, we are going to treat the mice, not the genetic deletion, but the mice when they develop tumors, you treat that with MIF inhibitors or anti-MIF antibodies. So those experiments are going on. Just 2 slides about microRNA and cancer, and the reason is that several chronic inflammatory products, including reactive species like nitric oxide and several cytokines, can regulate several different microRNA that functions like tumor suppressor or oncogenes. And this is a simplified cartoon showing that there are several cytokines that regulates a number of different microRNA, for example, 155, 21, 146, that are involved in several different cancers, including several chronic inflammatory diseases like ulcerative colitis and chronic pancreatitis. So in summary, that it's the overall balance of inflammatory signal that determine the outcome in tumorigenesis, whether it is initiation, progression, or metastasis. So lastly, I would like to summarize what I said earlier. Many chronic inflammatory diseases increase the risk of cancer. Inflammatory and immune mediators contribute to different stages of tumor development and progression. Nitric oxide can activate p53 response pathway and may also induce oncogenic p53 mutation in ulcerative colitis. Increased expression of inflammatory mediators, example MIF and nitric oxide, enhances disease aggressiveness in pancreatic cancer, and targeting MIF and nitric oxide induced signaling increased survival in mouse model with lethal pancreatic cancer. So that provides a proof of concept that it can be further moved towards more preclinical studies and then to clinical trials. And the highly complex interaction of immune and inflammatory signaling determines the overall effect of inflammation and tumorigenesis. I will stop here. Thank you very much for your attention.
Perhaps you could elaborate on the changes in the microRNA. How quickly does that happen?
Yeah, so
what's the magnitude of the change?
Yeah, so in our hand, when we exposed the cells to nitric oxide, different pancreatic cancer cells, you see the expression of, so we did a microRNA array analysis. So in 48 hours, we see the differential expression of a number of different microRNA, including 221 and several other microRNA. So within 48 hours. In the mouse situation, in the mouse model, where you have deletion of NOS2 or MIF, there also you see differential expression of microRNA. But we are doing a time point now by sacrificing the mice after, for example, 4 weeks and 8 weeks, when they start showing the pre-neoplastic changes. So we can't answer to that question in vivo, but in vitro, within 48 hours, we see changes.
So it's the same course basically as EMT?
Yes. Yes. Yeah, yeah, yeah. So MIF functions by its binding to several different receptors. So there is a receptor complex on the surface, CD74, and then CD44 inside the cells. So it binds to CD74, but CD44 is important for transmitting the signaling of MIF. MIF can also come inside the cell through micropinocytosis, and once it is inside, it can activate ARC signaling pathway. It can activate your COX-2 pathway by enhancing phospholipase A and arachidonic acid metabolism. It can also enhance NF-kappa B through that signaling. Also, MIF can bind to CXC2 and CXCR4 receptor. So CXC2 and CXCR4 are also important in many different cancers, including pancreatic cancer. And in fact, its binding to CXCR4 can lead to the chemoattractive function, which I mentioned in the beginning of my talk. Yes.
So is the mutant RAS, the KRAS, having direct transcriptional effects on
MIF?
In the epithelial tumor?
We don't know. We don't know, but mutant KRAS definitely induce inflammation, and you see the generation increased expression of NOS2, whether it is going through TNF-alpha, other cytokines, IL-6, IL-1 beta, or direct KRAS, we don't know.
Okay, that'll do it, Parag.
Great, thank you very much.
Terry, I see that
That's an old
the
handout.
was 2 years ago. Maybe it's my hand I didn't send.
No, you just sent it too late.
Okay, I sent it too late. Yeah, okay. Thank you, Terry.
Okay, so as Tom mentioned, your handout's not going to correlate exactly what is talked, but it should be similar. So Tom received his PhD from biochemistry at the University of Virginia, subsequently did postdoctoral studies at Yale University, and he joined NIAID in 1988, became a senior investigator in the Laboratory of Host Defenses in 1996. The title, Knox Family NADPH Oxidases. Tom.
Thank you, Terry. So we're going to switch now from the NOS enzymes to the NOx enzymes. The NOx being a contraction for NADPH oxidases. And I see a number of interesting parallels between tumorigenic effects on NOS and the NOx enzymes as well. And I'll bring out some of those interesting parallels in the later part of my talk. So the history of this field concerns this remarkable feature of phagocytic blood cells, such as neutrophils and monocytes. They have this remarkable capacity for generation of large amounts of superoxide anion, which is a free radical formed by donation of a single electron to molecular oxygen. And the superoxide anion is very unstable. It's a free radical, plus it's charged, and it will rapidly dismutate either spontaneously or catalytically to form hydrogen peroxide. And in phagocytic cells such as neutrophils, you have a peroxidase, myeloperoxidase, that uses hydrogen peroxide and chloride to make hypochlorous acid or Clorox bleach. And the hydrogen peroxide and the Clorox are both very potent bactericidal agents. And so neutrophils are famous for this so-called respiratory burst, which is a rapid consumption of molecular oxygen that actually has nothing to do with respiration, but has everything to do with this enzyme complex called NADPH oxidase, which is a multi-component enzyme that assembles from its components that are usually maintained in a dissociated latent state. So with the event of engulfment of bacteria, you have generation of these microbicidal oxidants that kill bacteria. And there are patients who don't do that. They have a condition called chronic granulomatous disease, and they suffer from remarkable susceptibility to what we would normally consider low-grade pathogens. Things like Staph aureus and Aspergillus mold can be very serious pathogens in these patients. And my colleagues in our institute, my lab chief, Harry Malik, and John Gallen have accumulated the most, say the largest cohort of patients with chronic granulomatous disease here at the NIH. And we now know that the genetic lesions that give rise to this defect in this respiratory burst oxidase can affect any one of these 6 components of this enzyme complex. The core of it is this transmembrane flavocytochrome, which is membrane embedded, translocates from secondary granules and which fuse to the nascent forming phagosome. And you have the other supportive cytosolic factors, GTP-RAC, and phosphorylated other phagocyte oxidase factors that come together and give rise to this respiratory burst during the event of phagocytosis. So this oxidase has been known for decades, and we now know in this post-genomic era, since the late 90s, that this enzyme is but one representative of a whole family of NOx enzymes, which are now summarized here in this schematic slide. So the GP91-FOX core component, the flavocytochrome of the phagocyte system, has been renamed as NOx2. That's a long story on its own. But the first of the non-phagocytic enzymes is the NOX, now NOX1. And we have NOX3, NOX1, 2, 3, and 4. They all have very similar structure. They all share in common conserved sequences that are required for binding of NADPH in the cytosolic reductase domain, a flavine binding center, and two membrane-embedded helical segments that have conserved histidines that coordinate two heme moieties, so that you have this, this enzyme that takes electrons from cytosolic source, passes them through the membrane, donates them to molecular oxygen in an extra cytoplasmic compartment, such as the phagosome. So, as mentioned here, these other NOX enzymes are very similar in structure, and their homology to the phagocytic prototype is on the order of 25 to 55% identical sequence. Then we have this NOX5 enzyme that has an amino terminal extension that contains these calmodulin-like calcium-binding EF hands, and this enzyme is calcium activated. And then we have the so-called dual oxidases, DUOX1 and 2, that also have calcium-binding EF hands, and then another amino terminal extension with another membrane-embedded segment, and an ectodomain with peroxidase homology. So you have NADPH oxidase and peroxidase domains in the dual oxidases. So just to give you an outline of where today's session is going, I'll start. Well, I'm going to emphasize that these other non-phagocytic oxidases are most abundant in epithelial cells, and they appear to all have roles in host defense, inflammation, and oxidative stress. And I'll start with an overview of the structure, their expression patterns, and functions, provide an outline of what's been learned from NOX mutations in both mouse and man. And then I will dwell on three separate topics, work that in our lab has highlighted functions of NOX1, NOX4, and DUOX in three different tissues. Here, colon is a prevalent oxidase in colon epithelium that has a lot of similarities with the phagocyte oxidase and is responsive to microbial patterns in the gut. In the case of NOX4, we and others have emphasized its induction by TGF-beta, and it appears that it has pro-fibrotic effects when overexpressed. And then in the case of the airway, the DUOX, we have studies indicating roles both in antimicrobial and pro-inflammatory responses. So as my earlier slide indicated, all of these NOX enzymes are electron carriers. They're membrane embedded, and they take electrons from a cytosolic source, NADPH. You get 2 electrons from NADPH. And these are passed in series to molecular oxygen, giving rise to superoxide anion as a primary product. And in all reconstituted or native systems where we overexpress these NOX enzymes, we can detect superoxide. And then in contrast, the dual oxidases with this peroxidase-like ectodomain, when we reconstitute these enzymes in transfected cell models, we don't detect this superoxide intermediate. And we and others are thinking that this peroxidase-like domain has more to do with peroxide generation rather than peroxide utilization. And I'll have more to say about that later. So these core NOx components, as I say, are flavoenzymes. They're the electron carriers. And we and others have found over the years that they're not standalone enzymes. They have other protein partners. In the case of NOx 1, 2, and 3, we have a requirement for GTP-loaded RAC. So these are all RAC-dependent enzymes. And we also have this NOx activator component or its homolog, P67-FOX, that bind to RAC in a direct fashion that's GTP-dependent, and that partner endows the enzymes with a GTP sensitivity. We have this other NOx organizer component, or its homolog, P47-FOX, which is a modular protein that links interactions with P22-FOX and the NOx activator components. And then we have the P22-FOX component itself as a common partner to NOx 1, 2, 3, and 4. And it acts as a stabilizer, a stable subunit of these heterodimers, and it has a role in the maturation of these enzymes. And then we have, in the case of the dual oxidases, we have something that acts like the P22-FOX. It is also a maturation factor, which has been called Duox activator. So just to illustrate this NOx P22-FOX interaction and its consequences in maturation and translocation, we have shown in this easily transfected model, HEK cells, the endogenous P22-FOX is detected in the endoplasmic reticular pattern condensed around the nucleus. And when overexpressing transfected 22-FOX, we see the same ER pattern. But when we express NOx 1 or NOx 3, we see that the P22-FOX then translocates from the ER out to the plasma membrane. So these cells have an outline of the plasma membrane staining for P22-FOX. And the cells that are not transfected, you see they're not staining as brightly for 22-FOX, and they have the ER-like pattern. So it appears that the co-expression of these NOx chains stabilizes higher levels of the 22-FOX in these cells. So they co-stabilize and they co-translocate, and then the enzymes are disposed in the plasma membrane. They deliver their reactive oxidants to the extracellular compartment. Now, similar thing goes on in the case of Duox and its interaction with the Duox activator. It's also a maturation factor, and for years, investigators have tried to detect Duox activity in transfected models, and it was only after Helmut Grasberger described these so-called maturation factors that are required to enable the exit of Duox from the ER compartment. And the two proteins go together, undergo Golgi-based carbohydrate modifications, and then you detect the two in a covalent disulfide linked complex on the plasma membrane. And only when both are co-expressed can you reconstitute H2O2 generation in these cell models. So here I've cataloged a variety of observations described over the years. And as I have alluded to earlier, the defect in NOx2 or any of its other supportive cofactors, mutations in any one of these can give rise to a defect in microbicidal activity in phagocytes and in a condition called chronic granulomatous disease. And in addition to enhanced susceptibility to infection, these patients have dysregulated inflammatory problems. They have autoimmune disorders, colitis, arthritis, and also some vascular phenotypes have been described. And this, in this case, turns out that CGD phenotype actually is protective against atherogenesis, where you no longer have macrophages with this ROS generator invading vascular tissue. You have diminished atherogenesis. So in the case of NOX1, people have appreciated its high expression in the colon since its first description in '99 by Lambeth's lab. And the other place where people have focused attention is in vascular smooth muscle cells. And the production of superoxide by NOX1 in vascular smooth muscle cells has a vasoconstrictive effect. It's the opposite effect of nitric oxide. So nitric oxide, as you I presume have heard earlier, is vasorelaxive, but superoxide is vasoconstrictive. So in the NOX1 mutant mice, several labs have described a blunted hypertensive response to chronic angiotensin II administration. And then more recently, work from Andrew Nish's lab has described NOX1 targeted in colon epithelial cells gives an altered or diminished wound healing response in the gut. And I'll show more slides about that later. The NOX3 has a very unusual expression patterns, particularly high in the inner ear and your vestibular canals. You have NOX3 expression required for making these bony structures that float around in the inner ear and touch the hair cells. And so defects in NOX3 or its other NOX3 supportive components give deficits in gravity perception and balance in mice. NOX4 is widely expressed, and people have emphasized its role in vascular tissue. Depending on the experiments or the experimenters, the labs, there's been some debate as to whether the reactive oxygen generated by NOX4 is beneficial or harmful. So some say that NOX4 supports angiogenesis, and the ROS generated from NOX4 can be adaptive for conditioning, and others say it's harmful. Still an open dispute. In the case of DUOX, you have recent work by Vandervliet showing that the DUOX mutant mice have a blunted allergic asthmatic phenotype, and this speaks to role in airway epithelial cells that I'll describe. And then work by Miklos Geist's lab has shown that urinary bladder contractions are altered in the DUOX1 knockout mice. In the case of DUOX2, you have developmental defects that reflect the critical role of DUOX2 in the thyroid gland for thyroid hormone production. So thyroid hormone production requires hydrogen peroxide to iodinate thyroglobulin. I'll say a little more about that later. Recent work by Ulrich Knaus's lab has shown that genetic variants of DUOX2 can affect very early onset inflammatory bowel disease. And work by Helmut Grasberger's lab has shown that the double knockout of DUOX activator 1 and 2, in those mice, they have a susceptibility to Helicobacter infection. Just as an aside, illustrate the phenotype of the NOX2 mutant mice. Mice with a NOX3 mutation have what they call a condition called head tilt. And the NOX0 mutant mice have something they call head slant. And these mice, they don't know up from down. And if you drop them in a beaker, they can't swim. They don't know up from down, and they drown unless you save them. Wild-type mice know which way is up to save themselves. And if you look in the inner ear, these otoliths or otoconia, these bony structures that touch the hair cells, are absent in the NOX3 mutant mice. So our lab is concerned with host defense and inflammatory disease, and our own work has started with the phagocytic oxidase in the late 80s. And then in the late 90s, we got into several of these other non-phagocytic oxidases, primarily in epithelial cells. And over the years, we and others have accumulated evidence to support the notion that most of these epithelial NOX enzymes are involved in innate immune defenses. And the sort of circumstantial evidence that we and others have accumulated over the years include their induction by pro-inflammatory cytokines, case of NOX1 in the gut, induction by interferon gamma or TNF-alpha, NOX4, uniquely inducible by TGF-beta, DUOX in airway and in skin is Th2 cytokine induced. DUOX2 is interferon gamma induced. Seems to be an antiviral oxidase. Several of these oxidases show responses to microbial pathogen or danger-associated patterns, MAMPs, DAMPs, PAMPs, and just to cite a few, you have NOX1 responding through toll-like receptor ligands or formal peptide, through formal peptide receptor, a bacterial peptide, NOX4 responsive to LPS, DUOX responsive to danger signals such as histamine, ATP, DUOX2 responsive to poly-IC as a mimic of viral RNA, or LPS, or moramal dipeptide in the gut. So we and others have shown how these NOX enzymes accumulate on epithelial surfaces, and in the case of DUOX, it's apically disposed such that we think these enzymes are involved in the barrier function at epithelial surfaces, and that the ROS, at least in some cases, is aimed outward into the lumen from the apical surface of epithelial layers. And then we'll mention some data on partnership of airway DUOX and lactoperoxidase, a well-known antimicrobial peroxidase, and also illustrate how NOX1 is functionally very similar to the phagocytic enzyme. So first part here on NOX1, just highlight here that NOX1 is responsive to these cytokines or microbial patterns. And these are features that have been recognized in phagocytes for years. And as mentioned earlier, they have dependence on RAC and these other cytosolic proteins. And the NOX organizer and activator proteins are detected by in situ hybridization within colon crypts. So here, it's an old slide from more than 10 years ago, in which we detect the messenger RNA with antisense, radio-labeled, and the silver grains are colorized in red. And you see that these NOX1 supportive proteins are seen in the colon crypts. And their structures are very similar to the phagocytic homologs. They have all the features required for RAC binding, for binding of the counterparts, the organizer proteins, through tail-tail interactions with these proteins. And you have these SH3 domains that dock to a cytoplasmic tail of P22-FOX. And lipid binding FOX domains that bind phosphoinositide lipids that are common features of both the NOX1 and NOX2 based oxidases. And we have shown by way of mutagenesis that if you make mutations in NOX activator or organizer proteins that disrupt these interactions, you have either partial or complete loss of superoxide production in these reconstituted cell models. So everything we know about the phagocytic oxidase assembly and activation seems to apply to the colon oxidase as well. And then work recently provided by Andrew Nish's lab has shown that the similarities to the neutrophil oxidase extend even further in terms of this formal peptide receptor. This is a chemotactic receptor in phagocytes, and now there's a newfound appreciation in colon epithelial cells in which they show that formal peptide receptor recognizes peptides shed by commensal lactobacillus species, and the lactobacillus stimulate through formal peptide receptor the activation of ROS production by NOX1, and this leads to enhanced proliferation and migration of colon epithelial cells that were studied in these scratch wound experiments that were actually done in whole mice. So they have this microscopic endoscope that they claim can make a reproducible scratch, and they studied the wound healing response and its dependence on lactobacillus bacteria. And it turns out that so here is the wound outline. The epithelial layer is highlighted in green, which is a beta-catenin staining. And you see over the course of 1 to 4 days, the re-epithelialization of this wounded area. And the wound healing response, day 2, and you compare with Hank's balanced salt solution versus exposure to lactobacillus, you see that the commensal bacteria promote a more rapid wound healing process. Here it's only partially healed, and here by 2 days, the lactobacillus promote better wound healing. And that wound healing is scored here with wild type exposed to lactobacillus. And that wound healing is compromised in the mice where they have the knockout of formal peptide receptor, or the knockout, this is intestinal epithelial cell targeted NOX1 knockout. We see impaired wound healing in those mutant mice. And in our own lab, we have confirmed and extended these observations with this transfected epithelial, colon epithelial tumor line, in which we can overexpress NOX1 and another NOX1 supportive protein, is peroxyredoxin 6. And we find that the closure, so this is a wound gap that at time 0 is almost as wide as this entire panel. And then 20 hours later, if we overexpress NOX1 and the wild type form of peroxyredoxin, we see the maximum wound closure. And if we have mutant forms of peroxyredoxin or NOX1, or leave out the NOX1 or leave out the peroxyredoxin, we don't see the same rapid rate of wound closure. This is shown here graphically. The closure and proliferation also is maximum when we have the overexpressed system. Change topics now to NOX4. The case of NOX4, it's a very different enzyme. It is not dependent on any of these cytosolic regulators such as RAC. And in many places, if you detect the mRNA and the protein, you also detect reactive oxygen. So it's a constitutively active enzyme, a standalone enzyme. And its regulation is primarily at the transcriptional level. And we and others have emphasized its inducibility by TGF-beta. When we first described this gene back in 2000, we actually called it RENOX. It's a renal oxidase. And then this other name, NOX4, was imposed. And And the reason we call it ReNOx is the expression of NOX4 is higher in the kidney than anywhere else, although it can be said that NOX4 is probably one of the more widely expressed of all the NOxes, and particularly high in many epithelial tissues and also in vascular tissues, high in endothelial cells, and then we have findings in cancer cells that I'll describe. So its expression here, detected by dark field microscopy, the in situ hybridization will give a signal in white, and you see that its highest expression is out in the cortical region of murine kidney, here shown at a higher level. And then in this colorized version, you see that the transcript is highest in the proximal tubules of the kidney. And its high expression in kidney has provoked many notions about its function, although there is no good evidence of a phenotype in the kidney as yet. I remind you that it's an electrogenic enzyme, and so it may have something to do with transport functions in the kidney. It is a constitutively active ROS generator. We have suggested it could serve as a ROS, as an oxygen sensor, and there's recent work by Nishimoto that has found that indeed the response to oxygen concentrations for ROS generation by NOX4 is within the range of tissue oxygenation levels. So it clearly could serve as a transducer where the ROS generation could serve as a reporter of tissue oxygenation levels. And there are indications of many ROS responsive genes are downstream of NOX4. And then we and others have looked at a number of receptor-mediated functions. Barry Goldstein has described NOX4 involvement in insulin receptor signaling. Aboud's lab is described in kidney angiotensin receptor signaling, and several groups, including ours, have looked at TGF-beta related signaling. Here we looked at NOX4 induction in hepatocytes that were infected with hepatitis C virus, or just as a surrogate, the core protein by itself will suffice to induce the expression and activation of TGF-beta signaling, which acts on NOX4, acts directly on the NOX4 gene promoter to enhance its expression, leading to excess ROS production, oxidative stress. Now, we showed that the excess ROS produced in hepatitis-infected hepatocytes could be blocked by neutralizing TGF-beta antibodies or by this dominant negative receptor, TGF-beta receptor, which acts early upstream of NOX4 induction. Or we could block ROS production with this dominant negative truncated form, or by silencing, we could intercept excess ROS production induced by a hepatitis infection. And so we have proposed that NOX4 could have a role in the excess oxidative stress that occurs with the progression of chronic hepatitis C infection, where you have, over the course of 30 years, you have early hepatosteatosis and diabetic symptoms, followed by fibrotic type disease, and then transformation and development of hepatocellular carcinoma over the course of decades. And people have appreciated for years that TGF-beta and TNF-alpha have major driving roles in the disease pathogenesis of hepatitis infection. And we would like to say that NOX4 has has a big part of that. So this business of fibrotic disease relates to this phenotypic transition, the so-called epithelial to mesenchymal transition, or EMT, which you heard about from the earlier speaker. The cells that, turns out TGF-beta is considered a major driver of EMT. And with this response, you have a loss of epithelial cell polarity. You have the dissolution of cell-cell contacts. The cells take on a mesenchymal phenotype of enhanced plasticity and migratory properties. And this EMT phenomenon has roles in normal physiological processes such as wound healing and differentiation. But we have emphasized its roles in fibrosis and cancer progression and metastasis. And several labs have highlighted TGF-beta based induction of NOX4 in a variety of tissues that are susceptible to fibrotic disease, such as in the lung. Danical has shown bleomycin induced fibrosis involves an induction of NOX4, and in kidney and in liver, as we've indicated with hepatitis C infection. And then we have other findings with other epithelial cells where TGF-beta induces NOX4 and affects migratory activities, such as in these two breast epithelial tumor lines. Here we have a wound closure. So these are scratch wound assays at time 0, and you find that with treatment with TGF-beta, you promote the closure of this gap within 24 hours. And likewise, in this metastatic line, it happens within 6 hours. And TGF-beta treatment of these two cell lines induces NOX4 transcript. None of the other NOX enzymes are affected by TGF-beta. So the wound closure appears to involve participation of NOX4. If we silence NOX4 or express this dominant negative inhibitory NOX4, we can interfere with the wound closure. And in studies that are published, cited here, we've looked at NOX4 induction by this transcription factor SMAD3, and we could block that with drugs or silencing. And the NOX4 induction will participate in wound healing, as I've shown, also fibronectin induction and FAC activation, an important cytoskeletal integrator that participates in cell motility. And we can block all of these EMT-related events by silencing NOX4. So we had asked, when comparing these two cell models, this one, the metastatic line that migrates faster, we asked, well, what factors would account for that enhanced migration? And it turns out that this tumor line has mutation in P53. And so we're talking about P53 losing its tumor suppressive functions and taking on other functions that are pro-metastatic or pro-migratory. This is described nicely in this article. And we have, within this context, we have looked at NOX4. And it turns out that the effects of wild-type NOX4 on TGF-beta induction are suppressive. So we have suppressed NOX4 in this context. The mutant form enhances NOX4 expression. So TGF-beta is pleiotropic effects that are modulated by mutations in P53. And here we say it involves NOX4. So the wild type suppresses NOX4 induction while mutant promotes. So we see here wild type suppresses, in this line, which is p53 null, the transfection of wild type suppresses the induction of NOX4 by TGF beta. This is with TGF beta. So we have this induction of NOX4, and it's suppressed here by wild type. It's enhanced or supported by the mutant forms of p53. And so this is looking at NOX4 levels. Here we're looking at migration or invasion. This is a Matrigel invasion assay. And we find that the migration is suppressed by wild type p53. And it's enhanced or supported by the mutant forms p53, 175 or 280. If we include the dominant negative NOX4 in there, we can suppress the migration phenomenon altogether. So NOX4 seems to be a part of this TGF beta and mutant p53 based migratory activity. And downstream of that, we have FAC activation. So wild type suppresses NOX4, mutant augments the NOX4 expression and these downstream events. And to appreciate that on a single cell basis, we show that even in this cell line that has a wild type p53, we overexpress mutant p53, we can override its suppressive effects. In these few cells that are transfected with the mutant p53, we get higher staining of their nuclei. This is the background cell wild type expression in the other cells. And in these very same cells, you'll note that the NOX4 expression is greatly enhanced. And in these cells on the migrating cell boundary, you see the NOX4 spread throughout the cell body. We have other studies we describe in this paper showing that NOX4 is required for phospho-FAC activation. So to summarize this section here, we're talking about the divergent effects of wild type and mutant p53 on NOX4 dependent migration, whereby, so we now have a growing list of mutant forms that are found in, these are some of the most common mutations in p53 in a variety of tumors, including pancreatic tumors, by the way. They all support higher NOX4 expression and cell migration, metastatic activity, whereas the wild type suppress that induction. So we consider NOX4 as a candidate target for maybe any metastatic drugs. So in this last section, describe studies on the dual oxidases. Remind you that these are dedicated hydrogen peroxide generators, and they're directly responsive to calcium. And they were first described in the thyroid gland. And there's a mouse model in which was in the Jackson collection, a mouse with severe congenital hypothyroidism. And Johnson et al. Have described a point mutation in DUOX2 that gives this profound phenotype. This mouse is, this mutant DUOX2 mouse is basically has a cretinism. So it's got all the hallmarks of thyroid hormone deficiency. They have greatly enhanced thyroid stimulating hormone levels in their serum, greatly reduced thyroid hormone levels, and they have stunted bone growth, overall growth, and they have this point mutation here that we have tested in our reconstituted cell model. Agnes Donko has shown that this mutant protein has a defect in its translocation from the endoplasmic reticulum out to the plasma membrane. And the role of DUOX-derived hydrogen peroxide generation in thyroid tissues is twofold. You have two peroxidative reactions involving partnership of DUOX2 with thyroperoxidase. the, the peroxidase needs hydrogen peroxide to take iodide to iodinate tyrosine residues on thyroglobulin. So you get the iodination of tyrosines, and then you have also peroxidative cross-linking of tyrosines to give an ether linkage. So you have a dityrosine. This is a precursor of residues bound to thyroglobulin that then get exported and released as the mature hormone. So you need both thyroperoxidase and peroxide from DuOx2 to accomplish this. And we now know, so that was, the first description of DuOx2 was in 99, and in the early 2000s, 2002, you have a Dutch group that has described point mutations in human DuOx2. We now have a growing list of mutations in DuOx2 or DuOx activator 2 that are linked to congenital hypothyroidism in man. Another animal model for DuOx mutations is actually the panda bear. So the question is, how does this carnivore survive on a meager diet of bamboo? And the answer is, it has hypothyroidism. So they have plotted here daily energy expenditure, and this report in Science, also described in the Washington Post last year. So the panda bear is on this curve here with all these low energy mammals, three-toed sloth and these desert rats. This dotted line is animals that have 37% of the energy utilization of these other animals. And they have hypothyroidism. This is the panda bear thyroid hormone T3 and T4 levels. This is some other low energy mammal, maybe George or Jeb Bush, another low energy mammal. And so it has, turns out it has point mutation, introduces a premature stop codon in the DuOx cDNA. So if you survey tissues with high DuOx expression, you'll find thyroid tissue is at the top of the list, skin, and then all these mucosal tissues. And you find in these epithelial tissues, the DuOx is condensed on the apical surface. Here in the gut of the pig, you see it's condensed along the apical surface of the gut lumen. So these peroxide generators are aimed outward in these mucosal tissues. And we looked at a variety of mucosal tissues. Miklos Geist in my lab years ago noted that every mucosal tissue where you have this antimicrobial peroxidase, you have detection of one or the other forms of DuOx. So starting with the thyroid paradigm, you have partnership of DuOx with thyroperoxidase, and we've proposed partnership of lactoperoxidase in mucosal tissues. And as you look in lower invertebrates, you also have DuOx isozymes that are partnered with different peroxidases. And here in the sea urchin, the ovoperoxidases use DuOx-derived peroxide to cross-link the fertilization envelope. And here you have the worms with DuOx defects have a cuticle defect, and they have problems in cross-linking of their extracellular matrix. And also in the gut of the mosquito, Carolina Barria's lab has shown partnership of gut DuOx with this immune peroxidase that cross-links matrix and forms a barrier in the gut of the mosquito. So the lactoperoxidase has been known for more than 50 years as being a microbicidal peroxidase in saliva, milk, tears, etc. And we had suggested that Duox was a source of peroxide supporting lactoperoxidase. We show its high expression in terminal ducts of salivary glands, in particularly high in the rectum. Also, we detect Duox and lactoperoxidase. And it's the generation of peroxide from this rat rectal gland is inhibited by this oxidase inhibitor. Turns out in the fly, if you silence Duox, the animals succumb to gut infections. You see higher colony counts in the RNAi-treated flies. They die. And you can rescue that by overexpressing Duox again. And in airways, we detect Duox transcript on tracheal epithelial layer. We detect lactoperoxidase in these submucosal glands, and then it gets secreted out to the airway surface layer that then partners with Duox-derived peroxide and thiocyanate to make hypothiocyanate, which is a microbicidal oxidant. And we could demonstrate this in primary human bronchial epithelial cultures, which we grow on an air-liquid interface. It takes 3 or 4 weeks before they reacquire a mature ciliated phenotype. So in this 100% confluent layer by day 25, you see 50% of these cells is staining positive for Duox, and these are the same cells that have cilia detected with antitubulin, and over the course of time we see enhanced peroxide generation coincident with enhanced Duox expression. And we use these cultures to show that we could kill airway pathogens that are oxidant sensitive when we've had the full system there: ciliated epithelial cells, Duox activators, lactoperoxidase, thiocyanate. If you leave any of these out, you have higher colony forming units. So here's maximum microbial killing. And if we add antioxidants or this oxidase inhibitor, we're up to higher colony counts. Likewise, in this case, ascorbate gives higher colony counts as an antioxidant. So we, over the years, have observed many functions for Duox: biosynthetic functions, matrix cross-linking, hormone biosynthesis, barrier formation, microbicidal activity. Others have described bacterial repulsion by peroxide and mucin production by Duox. And more recent studies, we and others have shown that redox signaling, inflammatory cell recruitment. So in airways, Van der Vliet has shown that a dust mite sensitized asthmatic model, mice, if you knock out Duox1, the inflammatory cell recruitment to airways is diminished in that asthmatic model. And Van der Vliet is also emphasized. Oh, here's another illustration of wound healing involving Duox in the zebrafish, where Neathammer has shown that a laser-induced wound here within a matter of a few hours leads to recruitment of inflammatory cells and marked with red fluorescent protein. And if they silence Duox here with these morpholino modified oligos, they show peroxide production here is down. This is a peroxide assay. And recruitment of inflammatory cells is down if you silence Duox in the fish model. And Van der Vliet has proposed in airways also. He's looked at chemical wounding and also allergen stimulation. He's proposing that ATP is an agonist that activates Duox production of peroxide, which then leads to wound healing or these other inflammatory responses. We have described activation of DUOX by histamine with these airway cultures. We showed that histamine receptor 1 agonist is responsible for induction of DUOX, derived hydrogen peroxide production, and it can be inhibited by these histamine receptor 1 antagonists. So to summarize this third and last section, the airway DUOX, we think, has a role in supporting lactoperoxidase-mediated microbicidal activity. I can also maybe inflict tissue damage with this more harmful peroxidase, and can also have effects in these asthmatic or wound healing phenomena. And the DUOX is stimulated by different activators or microbial agonists or these various inducers. So to sort of tie it all together, one of the overriding themes I hope that may have come across is that several of these epithelial NOx enzymes have they have distinct tissue-specific expression patterns, and in each of them you have wound healing or migratory cell phenomena that can involve different agonists, bacteria, danger signals, viral inducer, mutations in p53 that affect the induction of different NOxes, giving rise to cell migration or fibrotic phenomena that involve common players. And so I'll stop there and just get back to the overview of how various NOxes are involved in inflammatory or infectious disease, and entertain questions. Thank you. And if you want the updated slideshow, I can email it to anyone who contacts me.
Question? Can you comment on the evolutionary relationships of the DUOX and the NOx? Is there any relationship at all?
Yeah, David Lambeth has put out a nice article about 5 years ago. And so it turns out that the NOx enzymes have very ancient origins. It goes back even to the plant kingdom. So you might consider, for example, you drop an apple and it bruises, turns brown. You might consider that an inflammatory injury response involving generation of NOx-derived oxidants. Turns out that you have several of these NOx enzymes that are even in plants. The fusion of the peroxidase-like moiety with the other NOxes, I think, has occurred only in the animal kingdom. But the plant kingdom has NOxes that do have calcium-binding EF hands, so that has ancient origins. Yep.
Been around a long time.
Yes. So, I mean, we're talking about an ancient form of chemical defense, host defense, involving a generation of a variety of oxidants. Yep. There are also developmental roles for NOxes in the growth of root tips. So there are particular NOxes that promote elongation and proliferation of cells at the root tips. And they're also seen in multicellular fungal organisms as well.
Thank you, Tom. That'll do it.
Okay, thank you.